The second of our lectures on the change in chromosome structure. And in the first one, we looked at deletions, and now we're going to look at duplications. So duplication is exactly what it sounds like. It is a situation where some part of a chromosome is duplicated. It may be major parts of the chromosome, or it can be just a very, very tiny part. They're called micro duplications. But basically, you end up with a certain component. And again, we're not talking about euploidies or aneuploidies. We're not talking about gaining an entire chromosome. We're simply saying, in this case, EF, which is here, where that region of this chromosome was duplicated. So you end up with a longer chromosome containing all the original material plus some extra material. Again, just sort of how we might do this in a problem might look something like this, where we simply have a centromere, AB, and then we show it as having some extension. That you would recognize that as a duplication. There are a number of different types of duplications. A tandem duplication is basically what you're seeing here. EF is duplicated in tandem on the edge here. They're known as terminal duplications. There's all sorts of things. There's displaced duplications. In that case, EF might have moved up here closer to the centromere or something. It's still EF, and there was already EF here. It's just that it got duplicated and somehow it got moved somewhere else along the chromosome. These are known as displaced duplications. And then there are reverse duplications. Reverse duplications are fairly common. These are where you would have EF and then FE. So EF and then FE. So it would just simply be, instead of being tandemly replaced, it would be reversed. They would be facing each other. And these occur due to some loops and other things. Now, how would we get these? Well, I mentioned when we talked about deletions that there was a relationship between deletions and duplications. And one of the ways that you can get them, and there are multiple ways, but one of the ways you can get them is through unequal crossing over. So remember that when we synapse during meiosis, in prophase, these have to line up perfectly. Well, if they miss a little bit, and they often do, okay, and you have crossover, it's not going to be an equal exchange, and that's what's being shown here. Notice the centromeres are not perfectly aligned. So, of course, again, we're showing it as a flat unit, but it's three-dimensional in reality, but it's trying to illustrate as a cartoon. Here we would have A, centromere, B, cross over and pick up D and E. So we would lose some components there. On the other side, on chromatid 3 here, we would have A, centromere, B, C, cross over and pick up a C from the other homolog, and then D and E. We would have a duplication there, and that's what's shown here. Okay. We can show that if we do the first one, of course, the outer ones in this case don't change. The inner ones, we have the centromere, we have B, D, E, so we end up with an intercalary deletion. And at the same moment, we have A, centromere, B, C, C, D, E, which is this chromatid, which has a tandem repeat in the middle of it. Okay, and you can get other forms of these. So we would just simply have this form. It's a good way to look at both. Both of these can occur. One of the places we know there's very good evidence that we see this are in these things called microsatellites, which we've mentioned before. Again, you will be using in lab. These microsatellites are these little either repeats like this. This is a dinucleotide repeat of AT, AT, AT. They can be trinucleotide, tetranucleotide, or so. We've shown one before that was a trinucleotide repeat. We talked about these when we talked about Fragile X syndrome, and we said that there were these repeat units at the sort of the end part of the X chromosome, and the more of these repeats you had, the more likely you would have a break in a fragile point. Anyway, these occur, and they occur all over our genome. There's just thousands and thousands of copies of these various repeats. Here you can actually see that there's an AC, AC, AC repeat right above this one. Well, it turns out that these are places where the system tends to miss, okay, and it misses just a little bit. And when it does, you often get sort of a duplication going on here, either an extra AT on one side. It tends to be in units. It's not just one base, or you may lose AT or a couple of ATs on the other side. So our chromosomes, every time we go through meiosis, are actually growing and shrinking just a little bit. And that's what I told you when we first started all this. discussion of change in structure, I said I could guarantee that my chromosomes and your chromosomes, we had structural differences. And this is where the places I know that we have structural differences are in these microsatellite repeats. And they're very useful. This is an old way of doing it here, but kind of like what you might see in lab, but you end up with lots of heterozygotes that you can see, homozygotes, and this is more of a stylized version of it. So this would be a heterozygote for two repeats where one of them, what they do is they design a primer that will fit on one side of these repeats, and then a reverse primer on the other side. They amplify that up using PCR, and you just look at these, and you get the links, the various links of these. And if you have extra pieces in here, extra ATs, it'll be a little longer, so it'll take longer to go down the gel. If you have a shorter version, then it'll be a little faster, okay, and it'll go down the gel a little faster. So this is a heterozygote for maybe I don't know, doesn't matter, 180. This might be 80 and 40. This might be 60 and 40, etc. And then, of course, homozygotes look something like this. And you'll get to play with some of these in lab and practice with them, but they're very useful for distinguishing um, genetic variation that's out there in natural systems, as well as doing some CSI type things. All right. Now, it doesn't have to be just microsatellites. You can have true large gene regions. And in fact, quite large regions copied. One good example that you've looked at with the fly labs, if you notice, there's this bar called a bar eye. Well, there are duplications, tandem duplications of that bar region. And so if you have a wild type eye like this and you have a duplication and it's a heterozygous individual, you get a quite well reduced eye. It's about half the size of the wild type eye. If it's homozygous for this double bar like this, you get a much greatly reduced from even from that. So you're down about a third of what you were up here from the wild type. And then if you see the heterozygous for the double bars, okay, and you are a triple bar in this case, then you end up with this greatly, greatly reduced. So these are just tandem repeats that occur. And once you get either homozygous or heterozygous condition, they have direct effects on the phenotype. And this is from the book showing how you might get these, how you might have these unequal crossing over. And when you do that, it tends to be a region where this does occur quite frequently. You would end up with one chromosome having a duplicated tandem duplication. So this would be a bar. And then you have one that's missing that has a deletion for this. Okay, so a complete loss of it. And then, of course, if you have these two crossing over, you can end up with this double bar, which has this 3 repeats. So you can grow and change, and then actually you go back to a wild type on the other side. So depending which one of these becomes a gamete and actually fertilizes the eggs, you could end up with a wild type flies showing back up from a bar, or you can end up with this double bar condition that you might see here. Okay? So just illustrating that you can get these repeats, and they form very important relationships there. And they can certainly be important in human systems. Lots of times we have these repeats that end up causing difficulty because what happens is they'll end up being repeats maybe in the control region of genes. We haven't really talked much yet about gene regulation, but when you have a gene, you have a section, there's always control areas on the front. There's got to be a way to turn it on, turn it off, speed it up, slow it down. If the tandem repeat is there or a deletion is there, you may lose function of that gene, or you may overexpress that gene. This is just one example of a kinase that's involved in transmembrane movement in cells. And there's just an example here of a small tandem repeat. It's a fairly small insert repeat. It can be up to 400 bases or so. What's interesting about this is that this repeat is associated with acute leukemia, myeloid leukemia, at least has been well documented with that. It's on the inside of the cell, but this is the protein, of course, not the DNA. But the repeat is showing where the repeat would be in the DNA. And it results in some additional amino acids being placed into the protein. The larger this insert is, the more likely this acute leukemia will be an extremely problematic one. And so people have starting to develop tests for this leukemia that actually focuses on whether or not this repeat occurs. As well as the test, people are also looking at it as a target for how can we get, pharmaceuticals, specialized pharmaceuticals, to go in and attack these particular proteins so that you you can shut down this leukemic condition. So it's one of those interesting ones where you have clearly evidence of a tandem repeat, and the growing repeat, the larger it becomes a greater and greater problem. Okay? And repeats have not always been bad. There's some pretty good evidence that they've been important in sort of burst type things that occur in evolutionary time. We look at these species and they're going along, and all of a sudden you'll see diversification. And one of the ways, certainly, when you have that is a euploidy event where you double, as we said in the lower teleost, you sort of double the amount of DNA, and all of a sudden you have all these extra genes to work with. But it doesn't have to be that complicated. You can simply have duplications that occur. Human evolution has been argued that that's likely to be one of those areas where it has occurred. A series of papers, they go back almost a decade, but you have these papers about how we have the human genome is fairly unstable. We have a lot of these little small duplications that are going on, various kinds. We have displaced ones, we have tandems and all that. And it is thought that probably there's common ancestors started this process, and as we got into humans, we seem that we get these sort of common occurrence of these duplication events in our genome. So they occur quite commonly, and they've been good. They've provided ways to duplicate genes so that we could gene regions that we could specialize and use for various things over time. They've also contributed some problems such as schizophrenia and other things that are due to these duplications that have occurred. But if you're interested in it, there's some really nice reading that you can do about it as far as what's going on and the thought process of how unstable our genome actually is relative to these duplications and deletions and other types of modifications that can occur.